In this tutorial we're going to focus on genes. It will be very short and relatively simple. Remember that all of our coding, all of our genetic coding for who we are, and in fact for what every organism is on planet Earth, is encoded in the base sequence on DNA. So deoxyribonucleic acid DNA is, as this definition describes here, the chemical information database that carries the complete set of instructions for a cell. And the instructions are usually worked out via proteins. And it's the instructions on the DNA that codes for the lifespan, the maturity, the functioning, right through to the death of the cell. So it's basically genes that are the working subunits of DNA. Now, what do we mean by that? You've studied DNA earlier this year. You know its basic structure. As a double-stranded molecule, you can see it here. Double-stranded molecule joined with complementary base pairs, as shown here. So essentially what we mean by a gene is a particular length of a DNA molecule that carries information or a code that ultimately translates into a particular protein structure. So a gene is a particular set of instructions that codes for a particular protein or for a particular function. And I think that's a very good and concise definition. So a gene is a particular set of instructions that codes for a particular protein or for a particular function. If we look at the diagram to the right here, we can see this in a diagrammatic way as to how this gets worked out. So remember what we said a minute ago. A gene is a segment along the length of a DNA molecule, along the length of a chromosome. That gene carries coded information in its base pairs that is transcribed into what they call a primary transcript of messenger RNA. As we'll see a little bit later in this course, bits of that mRNA get chopped out to produce the final functional bit of messenger RNA, as shown here. That messenger RNA, as you already know, moves out of the nucleus into the cytosol and attaches itself to a ribosome. And on the ribosome, it is translated into a protein. So the information on the original gene ultimately leads to a set of instructions that decides what sequence of amino acids will be built. Ultimately, of course, this leads to a particular protein which will have a particular function. Could be a structural protein, could be a functional protein of some kind, but what that protein is determined by the set of instructions in the gene.